in tonight for this month's Science Cafe, presented to you by the UMass Med Postdoctoral Association. My name is Olivia Kojohari, and I'm one of the postdocs here at the UMass Chan Medical School, and I'm also part of the outreach committee of the UMass Med Postdoc Association. So we're really excited for tonight's topic of silencing genes by manipulating RNAs, which is a topic that some of you may have heard recently in the news. And in tonight's Science Cafe, our two speakers will talk to you about how the clever use of genetic silencing techniques can be used to prevent altered genes in our body from being able to cause cancer and other diseases. Before we get into introducing our speakers, I just wanted to quickly remind you that the views presented here, as well as in my presentation, as well as the presenters' presentations, are solely those of the presenters and not necessarily those of the UMass Chan Medical School. And also this session, as well as our other science cafes, are recorded and they are uploaded on our YouTube channel, which you can follow, and you can share the link with anyone who you think may have missed it and may want to watch it later. Also, if you have a question during our session, please type it in the chat box, and our Q&A moderators, Sneha and Priti, will ask your question on your behalf during the Q&A session. And with that, we will go ahead and get started. So if I can advance my slides. All right. Yes, and at the end, we will have a survey. So we will share the link with you after the talk. And if you would be please be so kind to fill it out and share your thoughts with us. That would greatly help us with our future talks. So with that, we'll get started and introduce our first speaker. So our first speaker is Dr. Dan O'Reilly. Dr. Daniel O'Reilly is currently a postdoctoral associate at the Khvorova Lab in the RNA Therapeutics Institute at the University of Massachusetts Chan Medical School. Dan did his PhD in Professor Masad Damha's lab at McGill University in Montreal, in Canada. Dan earned his master's in science degree in chemistry from the University of Southampton in the UK. It was there that he was first introduced to nucleic acid chemistry in the laboratory of Professor John Watts, working on the synthesis of peptide nucleic acid monomers. Currently, his research focuses on using siRNA to understand the role of the mismatch repair pathway in Huntington's disease. For his work, Dan received a Hereditary Disease Foundation scholarship. Dan's fascination with the field started at an early age when a family member was diagnosed with Huntington's disease. He now volunteers for the Huntington's Disease Association in the UK with their research panel, which advises on research grant proposals. So Dan, thank you so much for being here tonight and for presenting, and please take it away.
Thank you very much for the kind introduction. Let me just start by sharing my screen. Okay. Can everyone see my PowerPoint slide? Okay, excellent. So if I change the display settings to this, everyone should see my PowerPoint slide.
Right. Yes. Good.
Okay. So today I'm going to be talking about using a technique called RNAi, or RNA interference, to essentially silence disease genes. So a little bit about what happens in your cells is that there is this essentially manufacturing process where your DNA, which stores the genetic information in your genes, gets converted to an intermediary called RNA, and then this RNA gets converted into something called a protein. Now, the protein is essentially what performs all the actions in the cell. They do everything from metabolism, help transport oxygen around the body. They also even help stabilize the cell to keep it rigid. Now, essentially, this means that you can think of DNA as like a recipe book, where if you read the recipe book, you should be able to produce a nice cake like this. And this is what most of your cells do on a very regular basis, is that they produce nice, delicious cakes like the one on the right. Now, unfortunately, what happens is that sometimes there are mistakes in your DNA called mutations that result in problems downstream at both the RNA and then the protein level, which then causes disease. And so if we go back to this cake analogy, we can think of think of it as a mistake in the book, which then leads to, instead of a nice pretty cake, not so nice looking cake, as shown on the right, or as my dad likes to say, Dan's regular cooking. Now, the question is, how do we go about treating these diseases when there's a problem with in the DNA, these diseases? So to talk about that, I first have to tell you about DNA structure. So on the left is the classical structure of DNA with its 2 strands, and each strand is essentially made up of a combination of 4 different bases. They are A, T, G, and C. Now, what's great about DNA is that by using 2 simple rules, if you know the sequence of one of the strands, you can work out the sequence of its other strand or its complementary strand using A goes with T and G goes with C. And so, and vice versa. So if you put an A on, if there's an A on one strand, you put a T on the other. And if you put a G on one strand, if there's a G on one strand, you put a C on the opposite strand, and vice versa. So I've got a little sequence below to show you how this works. So if we start with a sequence that's TCGA, the first 2 bases on the next strand will be A because A goes with T, and then G because G goes with C, and then the next 2 will be C goes with, because it goes with G, and then T that goes with A. And so I just have a quick question right now, which is, I just want to make sure everyone understands this, which is, how do you go about, what are the 2 key rules of DNA? I can show them. There are 3 questions, 3 answers should pop out.
Yes, just give me a second. I'm trying to launch the poll and I'm having difficulty. Sneha, do you mind running the poll for me, please? Let's just see if Sneha is able to do it. All right.
Great. Just give one a few more seconds to answer. Okay. I think most people have answered. Okay, and it seems like most people have picked correct. The answer is C, which is G goes with C and A goes with T. And so I'll just stop the poll. And so what's great is that thanks to the Human Genome Project, which essentially found out the sequences of all your DNA and RNA, we can work out make complementary sequences that target specific regions of your DNA or RNA. So the question is, I've been talking about DNA and RNA, what is the differences and what are they? So really the big difference, essentially DNA and RNA are the same. They have the same 4 bases. DNA has A, G, C, T, as I said, and RNA has A, G, C, and U, but U is essentially the same as T for our purposes. Now, the one major difference between DNA and RNA is that DNA is double-stranded, whereas RNA is single-stranded, and so therefore it's easier to target using this complementary sequences that I've been talking about. So one way to target the RNA is using the short interfering RNA or siRNA for short. These are essentially 2 pieces of RNA that are bound together, where one of the strands is designed to be complementary to using those ATGC rules to the target RNA of interest. And what happens is when these siRNA get into the cell, they get loaded into something called RISC, which then unwinds the correct strand, targets the RNA of interest, and then cleaves that RNA. And then this stops protein production. Now, this process is called RNA interference, and actually, or RNAi, and actually Craig Mello at UMass Medical School discovered this and got the Nobel for it. Based on this process, there are 3 drugs approved, and there are many more in clinical trials. It's very exciting for siRNA right now. There's lots going on. Now, to talk a bit about my work, I have to switch gears from siRNA to rare diseases, which is Huntington's disease. So essentially it is a neurodegenerative disease, which means that the brain degrades itself over time. And this results in shaking, loss of like cognitive functions, and essentially results in death eventually. Now, Huntington's disease is caused by an expansion of a free base repeat which is the CAG repeat. And on the left, you can see that there is the normal Huntington gene with less than 30 repeats shown. And what happens is that when you get above 40 repeats, you start getting these loop-like structures. And then this is what essentially is cause for Huntington's disease. And I have shown here some MRI of patients where if you start on the left, there's pre-symptomatic patients, so before you get the disease. And then as you go further to the right, you see more and more degradation of the brain, of the regions of the brains. So there are two things that you should know about Huntington's disease. The first is that the amount of repeats that you start with is determines the age of onset. So if you start with 40 repeats versus 60 repeats, the person with 60 repeats will have an earlier age of onset than the one with 40 repeats. And also that it's been discovered that the CAG repeat expands over time. So if you start with 40 repeats, you will end up with some other unknown amount of repeats by the end of it. And this is a very important concept called expansion. Now, recently it was discovered that this expansion is caused by some actually genes called the mismatch repair pathway. And actually this pathway is a series of genes that's designed to protect you from mutations that occur and to stop people from getting cancers. So they're vitally important. But unfortunately, what happens when they see this loop-like structure like you see above is when this series of genes finds that loop, they then cause its expansion to occur. And this is not good for Huntington's disease because I just said that if you get more CAG repeats, it gets you an earlier onset of disease. So our lab is working on using siRNA to target these genes. In fact, one of the genes, MSH3, is highlighted. And what we've seen is that using siRNA, we can remove the MSH3 gene, so like get rid of it completely, and we stop the expansion from occurring in the Huntington's disease mouse model. Now, how this looks like data-wise is that if from these two graphs, what's happening is essentially we are measuring each additional CAG repeat over time. And in the top graph, each peak is a CAG repeat added. And in the top graph with no treatment, you get expansion. It just keeps occurring over the time of the experiment. But what we see below when we treat with our siRNA is at the time of treatment, we get complete blockage of the expansion. And this is really exciting as this could potentially prove a new therapeutic option for Huntington's disease because there's currently no treatments out there right now. And so with that, before I close, I just want to ask my final question, which is how many CAG repeats do you need to have before you get Huntington's disease? So if you could put the poll up, great. So just answer. Okay, great. I think most people have answered. I think we can stop. So yeah, most people have answered greater than 40 repeats. That's correct. With less than 30 and less than 20, those are actually considered within the normal range of having of the CAG repeats, so you won't actually get Huntington's disease. But with that, I'd just like to end my talk and thank everyone from my lab and collaborators and people who have backed us financially as well. That's been awesome. And thank you for your attention today.
Thank you, Dan. That was an excellent talk. I'll get started with the first question while we're waiting for more questions to come in. So what would be the normal role for the Huntington gene?
That's a great question. So it's a bit unclear fully the role. There's a lot of it has a lot more roles in developmental of the egg. It's very important then, but they think actually as an adult it gets less important, but it's still kind of unclear.
Interesting. Helene has the next question. So she says, hey, Dan, great talk. Could you tell us a bit more about the clinical trials that are going on with the oligos?
So there was 2 clinical trials based for Huntington's disease. 2 failed basically. And there are a bunch of reasons why. No one really actually knows why right now, is what I know from the companies themselves. They're looking into it. We hypothesize that the technology they're developing was actually to stop the mutant Huntington protein from being produced. And that was based on evidence, say like 10, 15 years ago, before this whole somatic expansion and understanding that the age of onset was linked to the CAG repeat number. And so we're hoping now that based on our studies, this is how we can create new therapeutic options.
Thank you. We've got another interesting question from Cassia. She says, hi, Dr. O'Reilly. Thank you for the talk. Why are you and your lab working with RNAi rather than CRISPR? And what are the pros and cons in Huntington's?
That's a great question. Obviously, CRISPR is the end game of like, you can edit the gene and get rid of the problem full stop. The problem with CRISPR right now is that there are 2 major problems I see. One is off-target effects. So like understanding the precise number of other genes affected by CRISPR, I don't think there is a big enough grasp on that to really start editing, like actually editing humans in big ways. The second is delivery of the CRISPR machinery to the even just the brain. Trying to get that throughout the brain is difficult because you have to deliver the whole CRISPR machinery, whereas in the case of RNAi, it's produced in the cell. And so that's how we can take advantage of that. And also with RNAi, it's more temporary than the CRISPR. So if you want to stop a drug, you can just stop injecting the drug, whereas with the CRISPR, once you've edited, that's it.
That's interesting. Thank you. Next, we've got a question from Bao. He says, hey, Dan, so less than 30 repeats are normal. What is the function of the repeat? Which is always an interesting question in an expansion repeat disease. And if the RNAi removes the targeted sequence, are there any side effects?
Great question. So yes, there are side effects from removing the targeted sequence because even though we design the sequence to be very specific to that RNA, there's always off-target sequences that are affected, and we have to understand that. And that'll be part of the process that we do moving this work forward. And then in terms of the CAG repeat role, that's a great question. I don't think I've seen any great data really understanding exactly the role of the CAG repeats in the protein. The problem with this kind of like proteins is that they aggregate really heavily. And so like doing any structural studies on the protein, etc., is very, very difficult.
Thank you. There's another interesting question from Bao. He asks, what are the mechanisms of this expansion?
So essentially, there is the mismatch repair pathway comes along, and they scan the DNA and they find the loop, and then they cleave it at specific positions. So if I go back to the slide, so like they can cleave it at specific positions on the DNA that results in expansion or not expansion. And because they see a loop, this almost like misleads the mismatch repair pathway to expand it instead of fixing it or removing it. So that's the mechanism.
So just to follow up on that, so this is basically during a replication, right?
Yes.
So in terms
no, actually, this is during transcription.
Oh, okay. So it happens at the RNA level.
Yeah, so the mismatch repair pathway and the expansion is shown to occur not necessarily during replicate. It does happen during replication, the mismatch repair stuff, but the Huntington specific stuff and the expansion of the gene is a transcription dependent series of events.
That's very interesting because that means that it can happen in neuronal cells as well.
Yeah, exactly.
Okay. So Revati has a question. He'd just like to know more about what are the diseases related to RNA.
As in, there are lots of, as in what diseases are caused by RNA. There are loads, like most genetic diseases have some level of, they all have, if you have a problem in the DNA, you are going to have a problem with the RNA as well. Now, whether that actually RNA is toxic or causes a disease, that's unknown. And in some cases in Huntington's disease, they actually think the RNA itself is also toxic. But most genetic diseases have some issue in the RNA as well.
Right. Yeah. It's quite a big field with RNA-related disorders.
Exactly.
Karen wants to know whether you're afraid that with this treatment, it could lead to cancer.
That's a great question. Yes, that is so MSH3 is not associated with brain cancers. And actually, it's known that if you get rid of it, other parts of the pathway can take up its role. That being said, what happens when our drug leaves the brain and goes to the rest of the body is something that we're very keen to look at. And so we will be looking at like our mice and whether they get more mutations due to our treatment of these siRNAs.
Thank you. Paurav says, cool talk, Dan. And he'd like to know which complex domain has been targeted such that the expansion can be stopped.
So right now we have MSH3 that has been actually shown with siRNA that we have data for that can stop it. There are other parts of the domain that I'm showing, MLH1 and MLH3. There are other types of data, not with siRNA, where they actually genetically knock out the whole gene. And those have been shown to block expansion. So we're hopeful that with siRNA, we can do the same thing.
Thank you. So Emanuela has the next question. She says, wonderful talk, Dan. Congratulations. What other diseases are you aware of that have been targeted with siRNA, not necessarily approved drugs?
So there are plenty of diseases out there. There's people in our lab working on Parkinson's disease, Alzheimer's, ALS, these types of diseases. They're all working on those kinds of diseases.
Thank you. I think that's all the questions that we have.
Can I ask something quickly?
Sure.
Yeah, it's wonderful talk. Thank you. So I may miss that. So where in the gene, which chromosome, like where specifically this CAG repeat occur? Can you clarify, like in the regulatory region or?
The Huntington gene is on the short arm of chromosome 4. That's where
chromosome 4. Oh, so it's always in only one chromosome.
Yeah.
Did that belong to a gene?
Yeah, it's in the Huntington gene.
Oh, yeah, Huntington gene. Is that like in the regulatory region or it's not?
That I am unsure about.
Okay. Thank you.
That's a good question, and I will look it up after this.
It's a great talk.
Thank you.
Thank you, Dan. That was an excellent talk, and thank you so much for the discussion as well.
No worries.
We can now move on to our second speaker.
All right. Thank you, Dan. That was excellent. So I will now share my slides again. Let's see if I can do that. Okay. All right. Can everyone see my slides?
Yes.
Yes, perfect. Okay, great. Well, thank you again, Dan, for such a great talk, and thank you everyone who submitted your questions. That was a great discussion. Our next speaker is Samantha Sarli. Sam received her bachelor's degree in behavioral neuroscience from Lehigh University. Currently, she's a PhD student in the lab of Dr. Jonathan Watts in the RNA Therapeutics Institute here at UMass Chan Medical School. In the Watts lab here at UMass Chan, she works on ways to improve the safety and activity of oligonucleotides for use in treating brain tumors and other neurological diseases. For her talk, Sam will discuss how silencing virtually any pro-cancer signal of interest can be achieved by using synthetic nucleic acid compounds called antisense oligonucleotides. Sam, thank you so much for being here and for sharing your research with us tonight, and the floor is yours.
Thank you. Let me share my screen. All right, let me just switch to laser. Okay, thank you so much for joining us in this discussion tonight. I think that there will be a lot of parallels between Dan's presentation and my presentation, so feel free to ask questions afterwards. But yeah, so I'm going to be discussing brain cancer and how we can use antisense oligonucleotides to kind of give it the silent treatment and silence certain kind of pro-cancer signal factors in these cancer cells and attempts to treat them. So let's start by revisiting a fundamental concept in molecular biology. Every cell in our body has a set of DNA instructions that it uses to carry out specific jobs for survival and for growth. So within our DNA are discrete units of hereditary information called genes, and these genes will get transcribed into this intermediate form known as RNA. And then this RNA then gets, through a process of translation, gets converted into a form called protein. And these proteins are complex molecules that carry out specific activities inside the cell that are important for things such as digestion, breathing, and even cognition. So this entire process from DNA to protein is known as the central dogma of biology. And so to make an analogy, Dan and I had a similar train of thought when we were thinking about a cookbook, but so if you think about the DNA as this instructional set, and then you have the RNA as kind of a handwritten recipe from the cookbook, and then this recipe can then be read and made into a hamburger, which represents the protein. The protein of the hamburger is separate from the protein inside the cell. So the central dogma holds true for both healthy cells and disease cells. And since DNA to RNA to the protein is a very linear process, if there is an error in the DNA, that error gets carried through to the protein, kind of represented here in this red coloring. And the protein ends up looking something like this, which something in this form is not able to do the same job in the way that it can in its original or intended form. So this dysfunctional protein is usually the source of the disease. And for example, there are many diseases like cystic fibrosis, Alzheimer's, Kruger, which is a human equivalent of mad cow's disease. These are all caused by either mutations in the DNA or dysfunctional proteins that can kind of wreak havoc and be detrimental to the body. So the question now is, how can we address these errors and what is the best way to address them? So we have a couple of options for trying to correct disease cells or prevent them from damaging nearby cells. First, we can edit the incorrect DNA directly to correct the mutations. We can also use small molecule drugs or antibodies to bind protein or prevent it from working in a way such that it's flagged for something like degradation. And then a third option is to target this intermediate RNA. So all of these systems are feasible as therapeutics and really depend on the disease you're trying to treat. But for my talk specifically, I will focus on the central form, which are the oligonucleotides. So antisense oligonucleotides, or ASOs, are a class of RNA-targeting compounds. In their simplest form, ASOs are short, single strands of chemically modified nucleotides. So either chemically modified RNA or DNA. Given that they are just nucleic acid sequences, they recognize and bind their RNA target via the base pairing rules that Dan so nicely talked about. Additionally, these ASOs are chemically modified so that they are more stable and more potent inside the cells, and they are also less toxic in the body. These are all things that you want when you're considering drug design. And then finally, because ASOs are made ASOs are made up of sequences, they can be modified quite readily to target virtually any gene of interest in most disease. So a different ASO can be designed very specifically for virtually anything or any disease, sorry. So for my first poll question, which will be up on the screen, I think, when do you think ASOs came about?
Just wait the 10 more seconds.
Sure. All right, looks good. So yeah, I think there's not really a majority, but 33% I guess guessed 1978, and you guys are correct. So ASOs have been around for quite a long time. They're not a kind of new field, but we are making a lot of new developments, which is why they're kind of becoming more prevalent currently. So one question would be, how do ASOs even work? And so if we look at this kind of the central dogma here, we have this ASO mechanism of action that once the ASO is injected into the body, they get inside of cells where they can bind their RNA target based on that sequence complementarity that Dan was talking about. And after binding the RNA, the subsequent duplex here formed by the ASO bound to the RNA recruits an additional factor called RNase H, which is not shown here, but this RNase H cuts the RNA specifically shown here in blue. And the ASO is then freed to find more RNA targets and go about binding them and then inducing cleavage there as well. So by cutting the RNA, you have no way then to make the protein. So the protein is never really a problem to begin with. So if we ask ourselves, why are ASOs really, unique in the sense of drug discovery? A major advantage of these drugs are that compared to small molecule drugs, they take a relatively short amount of time to make. So if we consider this timeline for, a small molecule drug, it can take upwards of 10 to 15 years to make sometimes, and then to get into patients. But ASOs are a lot quicker. For example, the ASO Melicin, for example, took about 9 months when it was when the disease gene was first identified to the day that the ASO was injected into the patient. And the reason the ASOs can be so quickly developed is because once you know the sequence of the gene you want to target, you can incorporate that sequence very easily into an already established chemistry that was optimized independently of the sequence. So you don't have to spend time kind of going through the chemistry and going back to the sequence and kind of, going between those back and forth. And this is in stark contrast with small molecule drugs that require much more extensive chemical optimization to achieve the desired drug activity and target specificity. So here the chemical structure dictates and can't really be modified separately of the other. So for the second poll question, I'd like you to guess which diseases have FDA-approved ASOs for treatment. Maybe 5 more seconds. Okay, so the answer to this poll is all of the above. And so let me stop sharing this poll for a second. Oh, I think I take this in exams, I see all of the above, I kind of assume that that's the answer, but for this case, it's the true answer. And you can see that, from this poll, you have an idea about the diversity of diseases that these ASOs can be used to treat. Currently, there are a total of 8 antisense oligonucleotide compounds that have FDA approval. In addition to these, though, we are seeing other ASOs rapidly enter the clinical pipeline for treatment. Dan briefly mentioned for diseases such as Huntington's disease, which is a hereditary neurodegenerative disorder, ALS, which is a motor kind of neurological disorder, and even more genetically complex diseases such as cancer. And so for the last part of my talk, I will present some of my own research in developing ASOs as an anti-cancer therapeutic. So cancer is a very general term that refers to a collection of over 100 different diseases, which are generally characterized by uncontrolled cell division and then invasion into surrounding tissues. The form of cancer that I'm interested in is a brain tumor called glioblastoma multiforme, or GBM. And this is a stage 4 brain tumor that has one of the lowest survival rates for pretty much any type of cancer in general. And then furthermore, the therapies for GBM have not been very successful in improving patient outcomes. I think it's been something like 20 years that we've actually had a drug that has, significantly improved the lives of these patients. So why are these tumors so challenging to treat? The first is that these tumor cells are very infiltrative, meaning they migrate throughout the brain as single cells, thereby escaping removal by surgical techniques. And second, these tumors are made up of many different types of GBM cells, which renders therapies like chemotherapy and radiation effective against some cells, but not all cells. And so the cells that are not affected by these treatments will continue to grow and proliferate, which is the case for most of the cases. And so from large sequencing efforts, we know that despite the diversity of tumor and tumor cells, there are specific recurring molecular abnormalities within the cell that drive growth and progression. Usually these abnormalities, which we call oncogenes, are specific to the tumor cells, and they are not found usually in the normal cells. So what we can do then is then design ASOs to silence these oncogenes to prevent that protein from being produced, which would otherwise normally support the tumor cell growth and survival from being made. So this will result hopefully in tumor cell death. And then an additional note to this is that oncogenes are usually found only in the tumor cells, as I mentioned. So even if the ASOs get into the normal brain cells, we don't have to worry as much about these compounds having, negative effects on these cells. And so finally, I just want to end with a little bit of data. So we have already tested some ASOs and their ability to silence a GBM oncogene called ATF5, which is a transcription factor that's important for the self-renewal of these cells. So we designed 20 ASOs shown here on the x-axis, and each number represents a different ATF5 ASO sequence. And these were all tested in the same GBM cells. And so from this screen of 20, you can see that we have identified 2 very potent sequences, this 11 here and 20 here shown in green, compared to the rest of them, and then other controls that we included to make sure that it was not an off-target effect. And then we made additional modifications. So here are the same green ones shown in this graph, but then we made additional modifications represented by the blue, purple, and gray bars. And you can see that we can further improve potency in silencing this tumor driver. And from a time perspective, it took us about 1 month to go from designing an ASO sequence for ATF5 or multiple ASO sequences to testing and identifying a lead ASO sequence. So our next step is to test these ASOs in GBM mouse models and see whether this ASO has any effects on the tumor. So that's the end of my talk. Thank you so much for listening, and I'm happy to answer any questions.
That was awesome, Samantha. Thank you so much. So I'll start with the questions, as more questions would be coming in. So do you know which one is more efficient, like comparing the efficiencies of ASOs targeting the glioblastomas versus a drug? Which one do you think is more efficient, or the efficiency is same for both the drug and the ASOs?
Yeah, so I guess I'll put this into context of a specific drugs. So temozolomide, which is an alkylating agent that is really used for these patients. So comparing that to ASOs to that drug, I think both have their pros and cons. One of the pros of like a drug, a small molecule drug, is that it can easily be administered, directly to the bloodstream, like via IV, and so it can cross the blood-brain barrier, where ASOs have to be delivered directly to the central nervous system itself. A drug can also, rapidly be taken up by more proliferating cells, where ASOs, there might be more chemical dependencies that, help it get into certain cells versus other cells. But I think with ASOs is the really, the crux of this is that you can make any sequence, and you can make many sequences and deliver them in combination to a tumor and, target many things simultaneously, whereas a drug kind of just non-discriminately targets certain processes of a cell. So I think with that specificity is what we really want, especially when we're dealing in the context of the brain, which we don't want to have too many off-target effects.
Okay. Thank you. I would like to ask more questions, which we have from the audience. And somebody's asking, what are the main differences in ASOs versus an siRNA? When would you pick one versus another for the particular disease?
Yeah, so this is a common question, and it's a good question. So the main differences between ASOs and siRNAs are that siRNAs are those double-stranded RNAs that work through the RNAi pathway. So they work through an endogenous pathway. ASOs just work on their own. So you don't have to maybe worry too much about the constraints of your chemical modifications that might interfere with the machinery of the RNAi pathway with siRNAs. But aside from that, they're pretty similar. And Dan can correct me if I'm wrong. And so when deciding to choose one or the other, I think both options are pretty viable. The only thing that might be a little bit different is that RNAi isn't, that machinery is not in the nucleus. So if your target's nuclear, you might want to go with ASOs. But other than that, I think both are fair game for therapeutics.
Okay, thank you. Next question we have from Sneha, and she's saying, very nice talk, Sam. Can we direct ASOs towards a particular tissue in the body? And
yes.
What determines their longevity? Yeah.
Okay, yeah. So the first part of the question, yes, we can and are actively trying to improve ways to target specific tissues. So an example, and this is more in the context of an siRNA, is that we have a GalNAc conjugate, which targets a specific receptor, I believe, that's expressed on the surface of hepatocytes. And so that GalNAc can then directly, help promote uptake of that siRNA to those cells in the liver. That same kind of idea does exist with ASOs, although, it's very hard to find conjugates that are very tissue specific. But we are working on that actively in the lab, things like glucose conjugates, folate conjugates, just specifically in the context of cancer. That might be, those are both really cool options that we're exploring now. And yeah, so it'd be really cool to have tissue specificity of these oligonucleotides. And then the second part of the question, I'm sorry.
Yes, the second part of the question is what determines their longevity?
What determines their longevity?
Yeah.
So that usually it's the type of chemical modifications that we use in these oligos. So these modifications, either on the nucleotide themselves or in the linkages, will help with nuclease resistance, will help, keep them in circulation longer. But yeah, there's not a hard and fast rule. Sometimes with different systems, different chemical modifications are preferred, whereas in other systems, others are better. So it's kind of there's some rules, but we don't know all of them.
Okay. So James is asking, do you have any criteria for which oncogenes you target?
So we are not too focused on which ones we target, because we can target any of them. I guess ones that have been shown in the literature to really be correlated with either the severity of the tumor, patient survival, or have been shown that when they are lost by these cells that they promote death. So those are the ones that we would really be focused on. But of course, we're happy to test new ones as they are discovered as well.
Okay. Casey is asking, how frequently is ATF mutated in glioblastoma patients? And why did you choose to target this oncogene?
Okay, so ATF5, it's not necessarily mutated, it's just overexpressed. And so by silencing it, then we can really kind of take all of its functions away from the cell. And so that reduces cell survival. And then why did we choose it, I think was the second question. There was a number of, the first is that it's a transcription factor. So those are really hard to target in cancer cells and just cells in general. So it'd be really cool to show a technology that could target something that's traditionally undruggable. But we just kind of we just picked something that had really strong evidence for its support and roles in GBM functions and started from there.
Yeah, I think Casey is also asking, how do you deliver ASOs in the brain? Like, how do they cross the blood-brain barrier?
Yeah, so the case of human patients, usually it's delivered via intrathecal, so into the spinal cord, and then it gets distributed via the cerebrospinal fluid throughout the brain and the spinal cord. And then in mice, we can deliver it directly to the brain via an injection into the fluid-filled spaces of the brain called the lateral ventricles.
Okay, thanks. Jennifer has the next question. What concentration of ASOs did you transfect in U87 cells?
Yeah, so that was at 50 nanomoles. So it's a pretty standard concentration for disease silencing.
And is it the same doses that you use in mice as well?
For mice, no, we usually, I'm not quite sure that, so it's hard to know the concentration, but we use about 30 nanomoles. So nanomolar and nanomoles. So it's about equivalent to see the same effect in mice.
Okay. And so there is another questions we have. Can we target viruses using ASOs and the siRNAs, for example, the SARS-CoV-2? And how about the bacterial, fungal, and parasitic infections? Can those also be targeted with the ASOs?
Yeah, that's a great question. And I think I would say that the answer is if it has a gene that is in, the RNA form, that we can target it. And our lab is actively working on projects related to COVID too. So yes, I would say that for other like bacteria and fungi, the problem would be a delivery. So can we get these ASOs into them? And so I'm not entirely sure if that is possible, but theoretically we would be able to, yes.
Okay, thank you so much, Samantha. So thanks everyone for joining in today. And now we'll end up for today's sessions.
Thank you so much again. Thank you, Sam, for the great talk. And thank you, Dan, for such an amazing talk. And if I will quickly share my screen again to just end with a final couple slides. Let's see. All right. Okay. So again, thank you for the speakers. Thank you everyone for tuned in and asked the questions. Please mark your calendars because we have another Science Cafe coming up, and our next one is on Thursday, December 2nd at 6:30 p.m. And also, we would like to ask you to please fill out the survey, which will be in the chat box. That would really help us tailor the events towards you, the audience, and feel free to suggest any other topics that you would like to see us cover in the Science Cafe. So again, thank you so much for tuning in, and thank you so much again, the speakers. We really appreciate you sharing your research with us. So thank you, everyone.